Hello everyone, my name is Ashley Palencia-Ortiz, and today I'm going to be sharing. Hello everyone, my name is Ashley Palencia-Ortiz, and today I'm going to be sharing my research on the synthesis of quarter period. Hi everyone, my name is Ashley Palencia-Ortiz, and today I'm going to be sharing my research. Oh my God. Hi everyone, my name is Ashley Palencia-Ortiz. I am an undergraduate in Dr. Schmenike's lab, and today I'm going to be sharing my research on the synthesis of quarter pyridine-based hexaaza-18 annuline. So my research mainly focuses on making molecules that can bind to metal ions. These molecules are useful in areas like material science and metal removal. In our research, we use a shift-based condensation reaction. The shift-based reaction essentially links together smaller molecules to form a ring-shaped structure. We also, in this reaction, use a strontium as essentially a template to help guide the ring formation. And our main goal for this project research is to create a more rigid structure and essentially understand how that affects metal binding. So for our objectives, we pretty much just kind of want to see if we can even synthesize this macrocycle, as well as study how rigidity can affect formation and binding, and eventually once we make that macrocycle, analyze the final product using mass spectrometry. So for our methods, we used a diformal quatropyridine and a diamine mixed in a pyridine solvent. We also added in a strontium as a templating ion to help organize the structure. Our reaction was done in a sealed vial and heated in an oil bath at about 100 degrees Celsius for about 8 hours. And during our reaction, a yellow solution formed, and after cooling, we saw a pale yellow precipitate. And then once we saw that precipitate in our solution, we filtered it, washed it with toluene, dried it over vacuum, and eventually weighed it out, and we got about an 89% yield. So down here in reaction products, these are some images that we took during our reaction. Down here, we see the initial yellow solution that we have when we added in the diformal quatropyridine, the diamine, and the pyridine. And then here we see the precipitate forming after the reaction cools. Here we see it being filtered out, and here we see the final yellowish crude product that we got. And these images essentially during the reaction help confirm that the reaction was actually progressing and producing a material. So looking at our results, the reaction did successfully produce material under our modifying conditions. The use of pyridine also helped improve solubility, and the sealed vial helped us keep the reaction conditions consistent. So over here, we see what is called a multi-spectrum. Each peak is pretty much different molecules in our sample. So instead of seeing one clean peak, we see two big ones and a couple smaller ones, which essentially tells us that we made a mixture. Some of these correspond to certain fragments of the macrocycle that we wanted to make. Some are just metal-bound species, and some are just other possible side products that we managed to make during our reaction. So up here we see a peak at 543, which corresponds to this molecule down here. This is not exactly what we wanted to make. The one thing that's pretty much missing is this bond from this amine here to this aldehyde here. But if we look back at this multi-spectrum, we see another peak, another major peak at 587, which corresponds to this molecule that's highlighted in green down here. This is essentially what we want our macrocycle to look like. And we see that we made some of it in our mixture. It's not entirely there. It's not entirely what we want to see, but seeing it on our multi-spectrum essentially tells us that we're taking the right steps to getting that clean peak on the multi-spectrum. So now that we're seeing two peaks, what does that really mean? So while we have the strontium in there, it will help form the macrocycle. It's the peaks in the multi-spectrum is telling us that the reaction is not fully selective. We can kind of conclude that there are competing reactions that are happening in that sealed pressure vial at the same time. Another big point is that the increased rigidity of this molecule down here um, may make it harder for the ring to actually fully close, and which is probably why we're seeing such a big peak here corresponding to this molecule. So even though we formed some of the target species, there's still a quite big mixture of other species. So, to wrap up my research, we managed to somewhat successfully synthesize this macrocycle using a safer and more controlled method. We can see that the reaction that we modified works, but the product mixture is not fully clean. So, some of our future work is going to focus on improving the reaction conditions to increase the selectivity and isolate the desired product. And down here is our paper from Thomas W. Bell, Frida Guzzo, and MGB Drew from the Journal of the American Chemical Society, in which we modified the procedure from. Thank you for your time.